Hey everyone, thanks for tuning into the Benchling launch event. My name's Andrew Dunn. I'm a pharma and biotech reporter at Insider. So it's my pleasure to introduce you to Dr. Kristen Hege, Senior Vice President at Bristol Myers Squibb and one of the pioneers of CAR T cell therapy. Thanks for joining me, Kristen.
Great, thank you so much. I'm Kristen Hege. I'm the Senior Vice President of Early Clinical Development for Hematology, Oncology, and Cell Therapy at Bristol Myers Squibb. I'm also a hematologist, a practicing hematologist at UCSF.
Great, and I'm really excited to spend the next half hour or so with you, Dr. Hege, to talk about really, challenges and promises around early drug development and some of the speed of getting research from, scientific innovation to an actual drug. And I think, a great example, and a great place to start is around your work in the space of CAR T therapies, which is, those are some of the most exciting programs right now in the field of oncology. And I'd love to start with some of the history around that and looking back at the period of early development for CAR T. Can you take me through sort of the origin story of CAR T therapies in your own work, going back to, basically the 1990s and where this idea began?
No, absolutely. I think many people think that the world of CAR T cells just sort of appeared in the last 5 or 10 years. But in fact, the early work in CAR T cells, as you mentioned, started in the 1990s. And, I had the opportunity while I was doing my hematology oncology fellowship training to go out and spend some research years at a small startup biotech company that had this, crazy idea that you could re-engineer the immune system to tackle, diseases with a high unmet need, such as HIV, which we were, fully immersed in back in that time in San Francisco, as well as cancer. So, I got started working in the CAR T cell field back in the early 90s. And the other thing people mostly don't realize is that this whole field really started in HIV, not cancer. The very first human CAR T cell trials were done in HIV. The very first trial was done in a group of what we call syngeneic twin pairs at the NIH, where an investigator, Cliff Lane, had collected a group of discordant HIV-infected identical twins. One had HIV and one didn't. That afforded the opportunity to collect healthy T cells from the uninfected twin, transduce them, create a CAR T cell product, and give it to the identical twin. Obviously, that was a very unique setting, and I was able to work with the NIH on that. And then we moved towards trying to make CAR T cells from HIV-infected patients, and reinfuse them back into the same HIV-infected patient. One would say today, maybe that was a hard place to start, but, we were young and naive and we didn't know any better. We really generated some very exciting data in the early days of CAR T cell therapy, how to grow these cells in the lab, the importance of CD4 and CD8 T cells, optimizing genetic transduction. But suffices to say, they didn't have the breakthrough results as the products of today. And it also coincided with the dawn of effective therapies for AIDS. When I first started in my medical training, HIV was a rapidly fatal disease. There were no effective therapies. And so we took this bold idea that we were going to try to just cure it with the immune system, because antivirals, the field was struggling to come up with effective antivirals. But then effective antivirals came along, and then the field got overly enthusiastic. I mean, there was a period when everyone thought we cured AIDS just by cocktails of what we call HART, highly active antiretroviral therapies. And the interest in exploring these kind of crazy, complicated treatments like CAR T cells waned, because everybody thought we cured AIDS with drug therapy. Of course, that wasn't true. But the field kind of pivoted then. We couldn't raise any more money, and our partners that were working with us on the HIV CAR T programs, walked away, and we pivoted from trying to develop CAR T cells for HIV to trying to develop CAR T cells for cancer.
Yeah, I find that fascinating as someone who covers the industry of if it's mRNA or the RNA interference platform, or really all these different technologies. They have these long histories where it takes several decades, and people don't necessarily realize or fully appreciate it when the medicine is actually in front of you. But I'm wondering, walking through that clinical development journey and the different hype cycles with, things come in vogue and then they go out of fashion. Was there ever a moment? I mean, what sort of brought it back as far as for
Yeah.
CAR T therapy when it found its place in cancer research?
Yeah, no, again, at this one small biotech company that was really the only biotech or biopharma company in the world working in this field, we launched a few trials in solid tumors. We, didn't see the breakthrough results that we've seen. And again, funding ran out and we moved on to other things. And the field of CAR T cells went back into academia, and continued to iterate. And, like many scientific breakthroughs, there's a cycle. And most true scientific breakthroughs seem to have a 25-year cycle time. Hopefully we can improve that. And you look back and say, well, what was the pivot point? Like, when did it go from failure to success? And with CAR T cells, I think we made a lot of early progress, as I mentioned, around the right T cell types, around how to grow them, around how to transduce them, around how to activate them. But we still weren't quite there until two things happened. One is we started to understand the importance of T cell co-stimulation. You can't just stimulate a T cell through one signal. You have to activate it through the primary signal and then co-stimulate it through a second signal to really get that robust T cell activation. So that was one new understanding. And the second was you needed to give this, what we call lymphodepleting chemotherapy, wipe out the endogenous T cells before you infused your CAR T cells. And that would, create a milieu that would favor the expansion of your CAR T cells. We call it homeostatic proliferation. And so it was really those two discoveries combined that took CAR T cells from ho-hum, crazy, esoteric optimists, just a few people in the world working on it, to the whole world standing up and taking notice. And really, there was always one seminal event. And with CAR T cell therapy, it was the December 2012 publication in the New York Times of this little girl with very refractory leukemia who was on her deathbed and got the first infusion at the University of Pennsylvania of a CD19 CAR T cell product, nearly died, and then her leukemia went in remission. She remains in remission to this day. That was a pivotal moment. Everybody, all the naysayers turned around and said, wow, like there may be something to this field. Maybe the immune system can kill cancer. And, the field has just exploded since then.
Yeah, and I want to get to, tying this into where we are now with some of the COVID response and also broadly just the challenges and promises around drug development, where the industry is headed. But I'm wondering, where do you think we are right now, both in terms of, I guess, if there's a hype cycle still, because I know there's a lot of cell therapy biotechs, and I just pulled numbers from, one recent report counts about 4,700 immuno-oncology drugs in development and about 6,200 active studies. So there's a lot of interest in this space now. And I'm wondering, what do you make of all of that? Is that a sign of promise? Do you think there's a little too much there?
Well, I've been a believer in using the immune system to kill cancer right from the dawn of my career. So perhaps I'm a little biased, but I think there's huge potential, and we've seen some of it, for engineering the immune system to kill cancer. And it's not just CAR T cells. Obviously, T cell checkpoint inhibitors have been a major breakthrough in cancer treatment, ways of just turning T cells back on after they've shut themselves down. So I think there's a reason for the hype. And, in the last few years, there are now 4 approved autologous CAR T cell products for different blood cancers. BMS is fortunate enough to be the only company to have 2 CAR T cell products approved this year, back to back, against 2 different targets, CD19 and BCMA, in 2 different hematologic blood cancers, lymphoma and multiple myeloma. So I don't think it's overhyped. I think it's the dawn of a new age. And that, just because it works in blood cancers, right, there's still some challenges and problems to solve in terms of bringing this breakthrough therapy to other cancer indications, notably solid tumors. But it is breakthrough. And in my, I don't know, 30 years in this industry, I can tell you that having the opportunity to work on the first in human clinical trial of a BCMA CAR T cell product in myeloma, where we are taking patients who had failed everything, who had no available therapies, who had, had transplants and maybe 16 prior lines of therapy, and you give them a single infusion of a CAR T product, and within 4 weeks, they eliminate all tumor cells from their bone marrow, and they obtain, deep, complete remissions. Like you don't see that very often in first-in-human oncology trials. So it really is transformative, and it's been very exciting to go from, the first patient dosed with a BCMA CAR T cell in 2016 to the last patient dosed in our pivotal trial in 2018, and then the first approval of a BCMA CAR T product in 2021. For me personally, because I had the pleasure of launching the partnership and shepherding that program from first patient to last patient to approval, it's been like a really amazing career highlight. And now I'm a treating hematologist. To think that my colleagues at UCSF can prescribe a CAR T cell product for their patients, like that's pretty exciting.
I'm wondering, was that a challenge in sort of going from scientific idea of CAR T and turning it into a commercial medicine, particularly something that can scale? How did you deal with that challenge, and are still dealing with the challenge of, scaling up and building in, scalable process and production methods?
Well, you can imagine, like, the launch of any truly disruptive therapy that is completely different than the therapies that have preceded it poses all kinds of new challenges for a field, which you tackle head on. And, moving from, say, a small molecule pill that can be made, hundreds of thousands of doses at a time and left, room temperature in a bottle on a shelf, like that's one end of the spectrum. And a lot of treatments, say for multiple myeloma, that's what they are. Moving from that to a patient-specific genetically modified cell therapy, that is manufactured and frozen and shipped and thawed and infused, it adds complexity. But these are not insurmountable challenges. I mean, take COVID vaccines, frozen vaccines made from RNA that have to be shipped everywhere in the world. That seemed insurmountable. And yet every CVS pharmacy in the United States seems to have COVID vaccines right now. So I always look at those things as like the solvable problems, the problems of supply chain and engineering and manufacturing and automation. Like, these are solvable problems. Like, fundamental problems of human biology are more challenging. And many of those, like, at least for these first generation of approved CAR T products, we have solved. Like, we have shown that you can engineer a patient's T cell to expand, proliferate, kill the tumor in the patient. So yes, of course, there are some challenges. I think what I get most excited about is, we've now dosed over 1,800 patients with CAR T cell products at BMS across 6 different diseases, 9 different manufacturing processes. And you're right, like, when you change the manufacturing process, you might change the characteristics of your final product, and that might change the way it functions or survives in a patient. But we have all this data now, so that we can interrogate what characteristics of the patient or the patient's tumor or their prior therapies impact the product attributes of the incoming apheresis material, and how do those product attributes affect the manufacturing of the final drug product, and then how do those drug product attributes impact how well that cell expands, persists, functions, the safety and efficacy profile. So we have this phenomenal data that we can interrogate and then use those translational insights to engineer the next generation of cell therapies to optimize, tweak it, whether it's tweaking the manufacturing process, modifying the patients, or actually engineering a different cell with different attributes. Like, we can start to fine tune the optimal CAR T product for different patients in different disease settings.
There's always a couple pandemic questions in every talk like this, and I'm wondering about comparing and contrasting the responses we've seen to the HIV crisis and the COVID pandemic, and in particular just your role and your involvement in both of them. I'm wondering what do you think has improved the most over those decades in terms of particularly the drug industry's response, but also I'd be curious to get your thoughts on the general public health response. And then are there what areas are still persistently challenging for the industry, even after all these decades and all these advancements scientifically? There's still things that are pain points and struggles to do.
Yeah, no, I think it's really important to go back to the history of HIV. Maybe I'm dating myself. I'm not that young anymore, but for better or for worse, I've had the opportunity to firsthand live through both of these pandemics. I went to medical school in the mid 80s. That coincided with the first cases of AIDS being reported, the discovery in 1983 of HIV as the cause of AIDS. And then you say, well, what happened after that? There was this new infection. It was caused by a new virus. How long did it take to get on top of that virus? HIV was, took 2 years to even figure out that HIV was the cause of AIDS. It took 4 years after that for the first marginally effective drug, AZT, to come along. And then 2 years after that for the first reasonably effective class of drugs, protease inhibitors. And then another 2 years before HART, highly effective antiretroviral 3-drug combination cocktails, cleared the viral load in patients, and AIDS went from a fatal disease to a chronic disease. And that transition, we were running trials in AIDS, like cancer trials, survival was the endpoint. Patients were dying of AIDS. We were trying to decrease the death rate. And then overnight, those trials were no longer relevant because no one was dying of AIDS anymore. And then you had to develop viral loads, and you had to get drugs approved based on decreasing viral loads as opposed to survival curves. So there was a transformation that happened during the AIDS epidemic, but I would argue in retrospect, it was painfully slow. And personally, I had a brother who was a homosexual. He got AIDS in the dawning of this epidemic. He died in 1995, the same year that AZT was approved. The year that protease inhibitors came out, but he had already developed an HIV-related brain lymphoma. It was too late for him to get a protease inhibitor. Had he lived another 2 years, he would probably be alive today because he would have benefited from HART therapy. So in terms of that sense of urgency that we need to develop drugs faster because literally patients are dying, waiting for new therapies to be developed and approved. That was when I transitioned from kind of a more academic physician scientist career in academia to a biotech career where I could work on, developing the next wave of innovative therapies for these serious diseases and try very hard to develop them faster. And we did make great progress during the AIDS epidemic. The FDA created accelerated approval pathways. It started the discussion that this has to happen faster. We need to cut through the bottlenecks. So then you fast forward to the COVID pandemic. The cases maybe November of 2019, and then, they identify the virus within a month. The whole world has the viral sequences within a month. The first vaccine trials are launched a few months later. We have emergency use authorization of the first vaccine using a brand new novel technology of mRNA within 12 months, and then full approval just last week within 2 years. That's unprecedented. But we still don't have a vaccine for HIV, I might add, after 25 years. So, lots of things enabled that acceleration. This was a disease affecting everyone in the world. So you captured the mind space of the world. And then, 25 years of science of RNA technologies, including things like liposome encapsulation, like had led up to the opportunity to catapult these RNA vaccines forward. And then, doing things at risk, manufacturing millions of doses before you know whether the clinical trials are going to be positive. Like that was a calculated risk that paid off. So I do think we've really accelerated based on advancements in, genetics, advancements in technology, advancements in data collection and analysis, advancements in manufacturing, supply chains. We've accelerated our ability to respond to these pandemics. But you mentioned what are some of the challenges we've seen? And I think we're all aware that it's not just about the science. I mean, I'd say the biopharmaceutical industry, this was probably one of the greatest advancements in the history of science to get these vaccines developed and approved as quickly as they were. But we've seen that there's all these other overlays, suddenly it becomes political, and all of the understanding of vaccine hesitancy, or even just, a public health system that wasn't really set up to deal with a pandemic in the United States. So I do think there's, misinformation. How do we control misinformation on the internet? So we tackled some incredible scientific challenges, and those uncovered these kind of societal challenges that we're still facing and for which we don't have complete answers.
Yeah, I think unfortunately those societal and really political challenges they seem more durable than sometimes the scientific problems that can be fixed over time. I'm also wondering on that idea of, this is more, I guess, a scientific challenge, at least, of those early months of the pandemic. And I'd be curious to get your thoughts, because I think this is finding that balance of speed and wanting to act, and then also making sure there's a level of rigor on the clinical trials. And there was a lot of studies, I think thousands, at least hundreds, probably thousands, that were launched in those first few months. And I've talked before with Janet Woodcock, who's now the interim commissioner at the FDA, and she's routinely over the past few years called the clinical trial system broken, and is a strong advocate for these things like master protocols and adaptive studies. And I'm wondering, how would you grade, looking back at those months, because the vaccine work obviously went tremendously well when it was sort of steered from the top down from an Operation Warp Speed like entity that could kind of guide and take the financial risk off. And at least on the treatment side, it seemed like we have some treatments now, but they're not necessarily transformative. And I'm wondering, what your observations are around there, of if anything rhymes with what happened around, the HIV/AIDS crisis, in looking ahead to future pandemic. Is there a way to do this better?
No, and I do think there's nothing like focusing the world's mind spread on one singular challenge to expedite things and to break through log jams. So I would say hundreds of clinical trials got started incredibly fast in terms of COVID therapeutic interventions. So I think everybody showed that we can do that. When, pushed against the wall, you can expedite all of this and get these things open rapidly. Now, again, that's the executional challenges, which we've proven we can tackle and solve the executional challenges. The fundamental biology of finding effective therapies for all of the different mechanistic manifestations of COVID is the hard part, right? 10 people can get COVID. One of them has no symptoms at all. The other one's in the ICU with diffuse pneumonia. And we don't completely understand why everybody responds so differently to the same virus, even if it's the same variant. So that's part of the, we need a deeper biologic understanding of the different manifestations of COVID in different patients with different makeups, different genetics, whatever it is, different comorbid diseases, so that we can target our interventions, for the right mechanism and the right patient. And we're not there yet with COVID. We kind of have some shotgun approaches, things like steroids, right, have been around forever. And then more targeted things like COVID antibodies, much more specific targeted therapies. Interestingly, doing different things. One's shutting down the immune response, the other's activating a COVID immune response. So I think we haven't made as much progress on therapeutics as we have on vaccines, because therapeutics are hard. But I think we will, because there's just so much focus on it. And hopefully it will be faster than the time it took to develop effective HIV antivirals, just because the science and the technology and everything else has advanced in the last 25 years.
Hundreds of millions of people already being vaccinated is incredible. And I'm wondering about the lessons learned from that you think could be applied to other areas in drug industry, and your own, perspective on how can, you accelerate, especially in early clinical stages, a promising drug. How can you shorten that timeline to market? When you look at, what mechanisms and tools were used with the COVID vaccines, does anything stand out to you as something, takeaways that can be applied to other areas in the drug industry?
Well, I think part of it is just the understanding that these breakthroughs don't happen in a vacuum. There had been 20 years of drug discovery in the area of RNA therapeutics and formulation that set the stage, primed the pump for the industry to respond incredibly fast to this COVID threat and say, maybe RNA vaccines is the fastest way to develop an effective vaccine against this threat. It didn't come out of a vacuum. It wasn't like RNA therapeutics got started in 2019. But all of that simmering science it sometimes gets forgotten in the background, but it sort of sets the stage. And then there's often one pivotal, either new demand that focuses the world, like a pandemic, or a slight tweak that takes something from less effective to very effective. And then in the case of the COVID vaccine, there was a lot of investment at risk. And we talk about it in our industry as you have must-win programs where you think they have such great potential, or they're going to satisfy such a big unmet need, that you're going to prioritize speed at the expense of cost and risk. You're going to spend more money, you're going to take more risk in order to shorten the timeline between discovery and approval. Other things that aren't quite so promising, you might not want to take that level of risk or spend that much money. You want to wait till you get some interim data that increases your confidence in the probability of success, right, before you make that next tranche of investment. I think with COVID, it was all in, right? All the investment, all in parallel, so that everything was moving forward. And as soon as you had positive trial results, you had millions of doses ready to be shipped. That was a impressive investment, and it paid out, right? I mean, if those trials had been negative, wow, that would have been a lot of sunk cost. Even then, I think we would have said that was a risk worth taking, because look at what was at stake. But it panned out, so it's a great story. How do we get smarter about early drug development? I've spent my whole career, for the most part, except for the approval of the CAR T product, in early drug development, because I love this innovative science-focused area of drug development where you're testing innovative new therapies for the first time. You know very little about them, and you're learning as you go, and you're pivoting. And we at BMS and previously at Celgene, we embedded early clinical development into discovery research, with the understanding that your early clinical development really needs to be driven by the science. You need to be hand in glove with your discovery translational scientists and understanding what are the characteristics of a patient that might impact response to this therapy, what enrichment factors, be they biomarkers around the mechanism of the drug, might select for an optimal tumor type to respond to any particular intervention. What are we going to measure in the clinic that convinces us that we've hit our target, not only in the blood, but we've hit it in the tumor, and that it's having an intended effect that we think is going to translate into, killing tumor cells, even if we can't measure that killing of tumor cells directly. So I think all of that deeply scientific focused drug development we hope is what is going to decrease that cycle time. Not only decrease the cycle time, but just be smarter and more successful. But so that by the time we have human proof of concept, like we're pretty confident that we know where this drug is going to work best, and with a higher probability of success in those pivotal trials.
I hear a lot from R&D leaders about this idea of prioritizing the kill projects, the tests that would, run that study where if that drug does fail, that's the end of the line for that project, and trying to find those earlier on in the development cycle rather than get to a phase 3 readout where, couple $100 million are invested in that drug and then realize it doesn't necessarily work or didn't pass the test. Do you see in the industry overall and at BMS more opportunities to run those types of, kill study is probably not the best word to use, but to run those make or break studies earlier and earlier in the cycle?
Yeah, I mean, you brought up a good point, which is we need to be smart about what we advance, and we also need to be smart about what we don't advance. Like both of those decisions are critical. And of course, we sort of simplify them down to go, no go criteria, but we try to establish those right out of the gates. Like before we dose the first patient with a novel product in a first in human study, we try as best as we can to define what are those first go, no go criteria prospectively, so that you're not like whitewashing your data after the fact, which is human psychology. You like to see the good in things. So that like being disciplined and objective, like if you don't hit a certain criteria, that you will kill a program such that you can shift those resources to something that actually meets the go criteria. So that's sort of the go, no go. And then it's like, what you referred to, we call them the killer thriller experiments, where like that one exquisitely designed experiment where the answer is going to be definitive enough that you're willing to either kill it or you're thrilled and you're going to advance it. So we do try to design those killer thriller experiments so that we can make the most informed decisions about what to kill and what to advance. And if you're at a company the size of BMS, where we have the good fortune of having such an exciting pipeline, I mean, we have small molecules, we have simple monoclonal antibodies, we have bispecific T-cell engagers and antibody drug conjugates. We've got endless variations on cell therapies, where, boy, you've got a lot of gene editing tools now. You can do a lot of things to, re-engineer a cell into whatever you want it to be. But you can't do everything. So you have to be able to prioritize, and we all strive to make the best decisions based on the science, based on the data. But then it is imperative that we collect the data that can best inform those decisions so we're not flying blind.
Yeah, and I wanted to finish with a question. I'm thinking of the audience here of Benchling's conference and launch of around, your own advice, practical advice for people in the industry who are in the earlier stages of clinical development and probably look at you and look at your career and trajectory and aspire to do something like that in the next, coming decades. What piece of advice do you have overall? And then I'm wondering specifically for people in clinical development roles.
It's exciting. Like, your careers are going to be incredibly interesting. And I think the cycle times, as we saw, like you look at the cycle times for HIV versus the cycle times for COVID, they're night and day. So the advancements that are going to be made in the next 25 years, probably 10x the advancements that were made in the last 25 years. So it's a really exciting time to be in research and development and, trying to create new meds for, incredibly important disease and to try to do it all faster. Have we optimized clinical development? No, there is plenty of room for optimization. And, there are still bottlenecks in areas where we feel like we're getting bogged down and you're just like, why can't we do this faster? Why does it take so long to make a protocol and activate a study and treat the patients and collect and analyze the data and write a report and send it all to the health authorities and review it? I mean, it's very tedious still. And I've got to believe in the next generation that a lot of the things that we do manually are all going to be digital. That many of the things that we did in person are going to be able to be done virtually. And COVID has been another like kickstart into that next future. I mean, I see patients every week at UCSF, literally overnight. We went from seeing patients in person in clinic to here's your 1-hour tutorial on Zoom. You know, all of your patients you're going to now see on video teleconference. Like that was literally overnight. And I can tell you, it was a painful overnight. And it took me a while to get my Zoom to work, you know, with the right access and not being interfered, you know, by my BMS passcodes. I mean, it was painful. But now patients prefer, for the most part, to have a Zoom telemed visit from the comfort of their own home instead of driving 4 hours to San Francisco, paying $40 for parking, you know, to see their doctor for 30 minutes. And then driving 4 hours home. So I do think as a field, we've gone more virtual in every way, and medicine is doing the same things. I think clinical trials, we're in the process, and it's been prompted by COVID, to shift as much of what we do in the clinical trial setting to virtual and remote patient visits, remote data entry, remote data review. And I think a lot of that is going to expedite and speed up the execution of clinical trials. But I do look to the next generation, the collective creativity of these minds that are not sort of haven't adjusted to the old way of doing things, myself included, to come up with really novel ways of doing everything that we do today faster and with higher quality and with less room for human error. But those are what I call the solvable problems. I would much rather see those problems, say, hey, I'll vote on the team that's going to try to tackle and solve those problems, as opposed to some fundamental problems of human biology, like tumor heterogeneity or the immune suppressive tumor microenvironment and how best to tackle it. You know, the genetic, the genetic fluidity of a cancer cell so that it can escape anything you do as soon as you do it. I mean, those are the really hard problems. Hopefully we'll solve these more executional, operational, data, virtual integration analysis, reporting challenges.
Yeah, that's a really helpful way, I think, of looking at it, those different buckets. And both are just fascinating from my perspective to watch and talk with people just like you about. So I wanted to thank you again, Dr. Hege, for taking the time here and sharing, some of your expertise and perspective going back with the pioneering of CAR T therapies and through COVID and where we are today. So thanks again for taking the time to talk with me. I appreciate it.
Absolutely. It's been my pleasure. Thanks, Andrew.